There are three stages in the process of transcription: initiation, elongation, and termination. Let's see how each stage works. During initiation, RNA polymerase binds to the DNA and recognizes a site called a promoter at the 3' end of the template strand of the target gene. When RNA polymerase finds a promoter, it breaks the hydrogen bonds holding the DNA strands together at the site of the promoter, and transcription begins. RNA polymerase does not bind to all promoters with equal affinity. This difference in promoter strength is one way that cells can control gene expression. The more strongly RNA polymerase binds to a particular promoter, the more likely that gene is to be transcribed. In general, higher levels of transcription lead to higher levels of translation, which leads to higher concentrations of that polypeptide. One strand of the DNA serves as a template strand. The RNA transcript is copied from this strand of the gene and therefore has an RNA sequence complementary to this strand. During elongation, RNA polymerase moves along the DNA, opening up a bubble in the DNA. As it moves, RNA polymerase adds bases to the 3' end of the growing RNA transcript. Elongation continues until RNA polymerase reaches a site on the DNA called a terminator. At this site, RNA polymerase and the newly synthesized RNA transcript are released from the DNA. Termination can occur through two mechanisms: self-termination and enzyme-dependent termination. In self-termination, the RNA sequence transcribed at the terminator causes the RNA to hydrogen bond with itself, forming a stem loop structure which essentially pulls the RNA polymerase off the DNA. In enzyme-dependent termination, a termination protein binds to the terminator and pushes RNA polymerase off the DNA.